Good day everyone, I am Emeline B. Casas from BSE 3A. Today, I am going to discuss about human cytomegalovirus. What is human cytomegalovirus? Human cytomegalovirus or HCMV is a beta herpes virus that causes lifelong infection in humans. HCMV is the leading infectious cause of congenital abnormalities in the Western world, affecting 1 to 2.5% of all live births. HCMV has a prevalence of 55 to 100% within the human population, depending on different socioeconomic and geographical factors. Primary HCMV infection is generally asymptomatic in healthy hosts but can cause severe and sometimes fatal disease in immunocompromised individuals and neonates. The characteristics of HCMV. The HCMV genome consists of a double-stranded DNA with approximately 230,000 BP. The genome is enclosed by an icosahedral capsid 100 to 110 nanometer diameter and 162 capsomers. Between the capsid and the virus envelope is a protein layer known as the tegument. The virus envelope is derived from cell membranes. At least 8 different viral glycoproteins are embedded in the lipid bilayer. The mature viral particle has a diameter of 150 to 200 nanometer. Like all herpes viruses, HCMV is sensitive to low pH, lipid-dissolving agents, and heat. HCMV has a half-life of approximately 60 minutes at 37 degrees Celsius and is relatively unstable at -20 degrees Celsius. It needs to be stored at least -70 degrees Celsius in order to maintain its effectivity. Human cytomegalovirus or HCMV is a member of the beta subfamily of herpes viruses. It is officially known as HH5 is a member of the beta herpes virinae subfamily of viruses characterized by slow replication and clinically asymptomatic infections in healthy individuals. Human cytomegalovirus or HCMV is a recognized cause of disease in the fetus, the allograft recipient, and AIDS patients. More recently, it has been recognized as a pathogen for those admitted to intensive care units, for the elderly, and for the general population. In brief, HCMV has the potential to spread in the bloodstream to all organs, but only produces overt disease if the viral load increases to high levels. This is normally prevented by a robust immune response so that the infected individual usually remains asymptomatic. HCMV intrauterine infection and can cause significant morbidity including low birth weight, hearing loss, visual impairment, microcephaly, hepatosplenomegaly, and varying degrees of mental retardation. Let us proceed to the prevention and control of HCMV. B cells also play a role in controlling HCMV infection and may also help prevent its transmission to new hosts as maternal antibodies can confer protection to fetuses in utero. The adaptive immune response contributes to the long-term control of HCMV. Also, it is important to wash your hands often. Avoid contact with tears and saliva when you kiss a child. Avoid sharing food or drinking out of the same glass as others. Be careful with disposable items, clean toys and countertops, and practice safe sex. Let's talk about ecological or medical impact of HCMV. People with weakened immune systems who get CMV can have more serious symptoms affecting the eyes, lungs, liver, esophagus, stomach, and intestines. CMV was strongly linked to socio-economic status, and CMV infection increased the annual mortality rate by 42%. Infection was associated with a near doubling of cardiovascular deaths, whereas there was no increase in mortality from other causes. These results showed that CMV infection markedly increases the mortality rate in healthy older individuals due to an excess of vascular deaths. And that's all about human cytomegalovirus. Now let's proceed to papaya ring spot virus or PRSV. What is papaya ring spot virus? Papaya ring spot virus or PRSV is a pathogenic plant virus in the genus Putivirus and the virus family Putiviridae, which primarily infects the papaya tree. It is transmitted between plants by mechanical activities like pruning and by numerous aphid species such as Myzos persicae. No seed transmission has been detected. The given photo is the left symptoms on leaves of papaya affected with papaya ring spot virus type P. Note the leaves with light yellow and green patches or mosaics and the deformed leaf on the left. Now let's talk about the characteristics of papaya ring spot virus. The virus is a non-enveloped flexus rod-shaped particle that is between 760 to 800 nanometer long and 12 nanometer in diameter. The first photo is a dark green stripes on leaf stalks of papaya caused by papaya ring spot virus type P. The second photo is the ring spots on papaya fruits caused by papaya ring spot virus type P. Papaya ring spot is a destructive disease characterized by a yellowing and stunting of the crown of papaya trees, a mottling of the foliage, show-stringing of younger leaves, water-soaked streaking of the petioles or stalks, and small darkened rings on the surface of fruit. Papaya ring spot virus belongs to the realm of Riboviria, kingdom of Orthornaviriae, phylum of Pysovericota, class of Stelpaviricites, order of Patatavirales, and family of Putiviridae, its genus of Putivirus and species papaya ring spot virus. Its synonyms are papaya ring spot Putivirus, papaya distortion mosaic virus, papaya leaf distortion virus, papaw distortion ring spot virus, papaw mosaic virus, and watermelon mosaic virus 1. Pathogenesis of papaya ring spot virus. Papaya exhibits yellowing, leaf distortion, and severe mosaic. It is oily or water-soaked spots and streaks appear on the trunk and petioles. The fruit will exhibit bumps and the classic ring spot. So, a severe isolate of PRSV has been shown to cause tissue necrosis. Cucurbit symptoms tend to be similar to papaya symptoms, including blisters, mosaic, yellowing, and leaf distortions. Let's talk about the prevention and control of PRSV. There is a known resistance to PRSV, although certain varieties are more symptomatic than others. There are four main methods of control for PRSV. First is the quarantine and geographic displacement. Second is the roguing and netting. Third is the cross protection. And last is the genetic modification of the host plant. Because PRSV is a non-persistent virus and is consequently transmitted to healthy plants by aphids within a very short time period, insecticidal control is difficult and impractical. Prevention through quarantine and geographic displacement of cropland is common and has occurred in Hawaii, the Philippines, and Brazil. Roguing, or the removal and destruction of infected plants, is a way to control the spread of PRSV. It is difficult to suppress the spread of PRSV, though roguing, because it is spread very quickly and effectively by aphids. Netting can also be used to prevent insect vectors from spreading the virus. Third is the cross protection is similar in practice, although not in mode of action to viral vaccinations in humans. A mild strain of PRSV is introduced into the host plant, which then develops resistance to virulent strains of the virus. It has been reported that papaya ring spot disease has limited papaya production in parts of Southeast Asia, Africa, India, South America, the Caribbean islands, and the North America. When papaya ring spot disease was first detected in Southeast Queensland in 1991, it ended commercial production within a few years. If the disease were to spread to the North Queensland production area, it is anticipated to have a similar impact on the North Queensland papaya industry and nearby local communities. Home gardeners can be severely impacted by papaya ring spot disease, with plants usually dying within 1 to 2 years of infection. Once infected, backyard plants become stunted, fruit production declines, and any resulting fruit have poor flavor and texture. And that's all about human cytomegalovirus and papaya ring spot virus. I hope you learned from this video lesson. Thank you for watching.